Good morning, everyone. So here I'm back with one more video. In this video, we will be starting with the review or the revision of nervous system. So specifically in this part of the video today, we will be doing the revision of the structure of the neuron, the synapse, and the various regions of the brain. The functions of the brain, I'll be discussing it in the second video. But in today's video, we will just be seeing the various parts of the brain that has been described by your textbook. So let us start with the structure of the neuron first. So let us have a look at the different parts of the neuron. The structure of the neuron you have already studied in grade 8 as well as in grade 9. This is a recap of the same description that you have already studied. So a neuron is the structural and the functional unit of the nervous system. Now the nerve cell or the neuron cannot divide after birth. It has lost its ability to divide after birth. Now, as far as the structure of a neuron is concerned, it consists of three main parts: the dendrites and the dendron, the cyton, also known as the cell body or the perikaryon, and third is the axon. So I repeat, there are three main parts of the neuron: the dendrites and the dendron, the cyton, also known as the cell body or the perikaryon, and the axon. Now, as far as the dendrites are concerned, the dendrites are basically the short and branch processes which extend out from the cyton or the perikaryon. I repeat, the dendrites and the dendron are the short and the branched processes. You can see the ones that are highlighted in green right now on your screen. They are short and branched, and they arise from the cyton. So there are several dendrites that arise from the cyton, and these are responsible for transmitting the impulse towards the cyton or the perikaryon. Now, let us shift the focus to cyton. So why do we call the cyton also as the perikaryon? Peri means surrounding and karyon means nucleus. I repeat, peri means surrounding and karyon means nucleus. So the cyton or the cell body or the perikaryon is the structure that surrounds the nucleus. Now, as far as the cyton or the perikaryon is concerned, it consists of several organelles, including the mitochondria. These green ones, the small ones that are highlighted, are the mitochondria. There are abundant mitochondria that are present within the cell body of the cyton. Then apart from the mitochondria, there is endoplasmic reticulum. You can see here, I'm just enlarging this image. You can see the mitochondria that are highlighted now. You can also see the rough endoplasmic reticulum that's highlighted in pink right now. And you also have the smooth endoplasmic reticulum that is highlighted in pink now. And apart from that, there are small granules of RNA called as the Nissel's granules. Okay, these Nissel's granules are responsible for performing protein synthesis. Now, the point on the cyton from where the axon is formed, that point is called as the axon hillock. I repeat, this highlighted part in green is basically called as the axon hillock. That is the region from where the the axon originates from the cyton or the cell body. Now, the axon along its length is wrapped by the myelin sheath. All along the length, the axon is wrapped by the myelin sheath. This myelin sheath is insulating material. It's a fatty tissue basically secreted by the neurolemma. So the neurolemma cells that surround the axon secrete the myelin sheath. Now, one interesting thing about the myelin sheath is that it is not continuously covering the entire axon. Rather, it leaves gaps in between. And these gaps that are left in between are called as the nodes of Ranvier. I repeat, they are called as the nodes of Ranvier. Okay. So now the mode of impulse conduction will be jumping, meaning the impulse will jump from one node of Ranvier to the next. So this type of conduction of the impulse is called as saltatory conduction. I repeat, it is called as saltatory conduction. So in saltatory conduction, the impulse jumps from one node of Ranvier to the next. I repeat, the impulse jumps from one node of Ranvier to the next. So this allows faster transmission of impulse rather than the impulse traveling all along the entire length of the axon. Now, as soon as the impulse reaches the axon terminals, we have these axon endings, which will be actually swollen in the form of a bulb. So there will be axon bulbs or the swollen knobs that will be present at the end of each axon. And these swollen terminals or the swollen knobs, I'll just highlight them here. I'll just turn this so you can see it. Yeah. You can see the knob-like structure at the end. So these knobs are basically called as the terminal buttons also. They are the ones that are storing the chemical called as the neurotransmitter, which is responsible for the release of these chemicals at the time of transmission of the impulse from one neuron to the next. So neurotransmitters are chemicals basically that are released by the axon terminal to help in the transmission of the impulse from one neuron to the next neuron. So example of a neurotransmitter molecule is acetylcholine. That's one of the examples of a neurotransmitter molecule. So this, in short, my dear friends, is about the structure of the axon. Now we shift our focus to the structure of a synapse. Now, synapse is basically the region of functional contact between the two adjacent neurons. Now, the dendrites of one neuron come in close apposition. Apposition means just opposite to the dendrites of the next neuron. I repeat, the axon of one neuron comes in apposition or in close contact with the dendrites of the adjacent neuron, but they are not physically in contact. Okay, remember that there will be no physical contact between the axon endings and the dendrites of the next neuron. So that gap in between the axon of one neuron and the dendrites of the adjacent neuron, that gap is called as the synapse or the synaptic cleft. I repeat, that gap is called as the synapse or the synaptic cleft. So now let us see the significance of the synapse or the synaptic cleft. So here you can see what is highlighted in green is the synaptic cleft. The synaptic cleft is the small gap between the axon terminal of one neuron and between the dendrites of the next neuron. Now the axon terminal or the axon bulb contains a lot of spherical structures which are called as the synaptic vesicles, which store within them the molecules called as neurotransmitters. So these chemicals that you see inside the synaptic vesicle are the neurotransmitter molecules. Now, the fusion of these neurotransmitters with the membrane of the axon terminal is regulated by the calcium ions. I repeat, it is regulated by the calcium ions. So these are the calcium ions that are now highlighted in pink. And these calcium ions will basically help in the fusion of the synaptic vesicles with the axon membrane. So you can see here, one of the synaptic vesicles has been shown to be fused here. Okay. And after fusion with the membrane, it will release the neurotransmitters. These neurotransmitters then bind to receptors on the membrane of the dendrite, that is of the adjacent neuron, allowing the sodium ions to enter in. So sodium ions, as soon as they enter in, they will cause depolarization. I'm sure you'll have learned about the wave of impulse, how it is transmitted. It's a wave of depolarization followed by a wave of repolarization. So in case if anyone wants an explanation for that same, I shall just give it here in brief. So suppose, let us say, for example, let us consider let us consider this to be the axon membrane. Now, suppose currently there is no impulse being transmitted, then the outer region of the membrane will be positively charged due to a higher concentration of sodium ions, while the inner region of the membrane would be negatively charged. As soon as there is an electrical disturbance, or if there is a impulse or an electrical disturbance, suppose in this region here, then in this region, there shall be a wave of depolarization that will be generated. Depolarization means there will be loss of polarity. The normal polarized state is positive charges on the outside of the membrane and negative charges on the inside. Now, due to depolarization, the sodium ions enter in. Now, this entry of sodium ions is basically because of the opening of the sodium gated channels. There are channels that allow the sodium ions to enter in. You will in detail learn about these channels in higher classes. But you can see here that due to the entry of sodium ions into the axon, the inside of the axon membrane has become positive, while the outer side has turned negative, while the remaining part of the axon membrane is still in the polarized state only. So the initial region, which has now undergone depolarization, serves as the stimulus for the next region to undergo depolarization. So this next region will now be undergoing, this region that I'm now highlighting will be the next one to undergo depolarization, whereas the previous region would repolarize. I repeat, the previous region will repolarize. So let us see how it happens, what happens here. So the region inside the box undergoes depolarization, whereas the previously depolarized region now undergoes repolarization. It again returns back to the normal polarized or the resting state. So again, in that resting state, the outside of the membrane becomes positively charged, while the inside is negative. While in the next region now, which is depolarized, the inner side is positively charged, while the outer side is negative. While the remaining region of the neuron is still polarized. It has not undergone any kind of changes as yet. Now, so this middle region that has undergone depolarization now serves as the stimulus for the next region. I repeat, the middle region, which is now depolarized, serves as the stimulation or the stimulus for the next region to undergo depolarization. So this is how a wave of electrical disturbance, which is called as an impulse, sweeps over the entire neuron. I hope the explanation is clear to all of you. So once a certain region becomes depolarized, that serves as a stimulus for the next region. And that way the wave of depolarization sweeps over the entire axon. But remember that this wave will only pass from one node of Ranvier to the other until it has reached the axon terminals, from where the neurotransmitter molecules help in the transmission of this wave of impulse to the next neuron. So I hope you all are clear with the structure of the synapse, as well as the transmission of a nerve impulse. Now coming to the structure of a nerve. Now, as far as a nerve is concerned, a bundle of several neurons, a bundle of several neurons together is called as a nerve. So you can see here, okay, I'll just hide these structures one by one so that you can actually visualize how exactly is this possible. I'll just hide all the wrappings around the coverings that hold together. You can see here, if I zoom in, this is the axon. So you can see several axons together are bundled inside. Okay, so these yellow dots that you see are all the axons. So a nerve is a bundle of several axons together. I repeat, a nerve is a bundle of several axons present together, packaged together. Now, as far as neurons are concerned, there are three types: sensory neuron, motor neuron, and inter or association neuron. A sensory neuron always carries impulses towards the CNS. A motor neuron always carries the impulse away from the CNS towards the effector organ. And the inter or the association neuron is always found within the CNS, that kind of forms a connecting link between the incoming sensory neuron and the outgoing motor neuron, basically. So the association of the interneuron is the one that will receive the impulse from the sensory neuron, analyze that impulse, and then generate an outgoing response or the impulse that will be carried by the motor neuron. So just as a neuron can be of three different types, likewise, even nerves are of three different types. The nerves are sensory, motor, and mixed, depending upon the type of neurons bundled together. So if you have nerve fibers that all are carrying impulses towards the CNS only, so then that bundle of nerve fibers, that is the nerve, is called as a sensory nerve, because all impulses are being carried from the sensory organ towards the CNS. The example of that can be an optic nerve. Optic nerve that arises from the eye carries impulses from your eyes, from the sensory part of the eye towards the central nervous system. Then you have the motor nerves. The motor nerves are the ones that will contain nerve fibers that carry impulses away from the CNS towards the effector organ, that is towards the muscle or towards the gland. So like, for example, the nerves that supply to the oculomotor muscles that help in the rotation of the eyeball. So that nerve carries impulses from the CNS to the muscles of the eyeball. So that's an example of a motor nerve. And then you can have mixed nerves. Mixed nerves will have both the type of nerve fibers or axons. That is, some of them carrying impulses towards the CNS, a few of them carrying impulse towards the effector organ. So such a nerve is called as a mixed nerve. So all spinal nerves are basically examples of mixed nerves. So that is about the neuron, the synapse, and the nerve. So in the next video now that I'll be uploading within a couple of days, we'll be studying in detail about the human brain. Thank you everyone for watching this video.